of the evolution meeting. I didn't have too far to travel, but it's been a great meeting so far. So I'd like to talk to you about a kind of side project I'm working on that's a continuation of some work I did in my master's, which is trying to decipher some of the radiations in blood-feeding flies. So the family is Humanipode, along with my advisor and another collaborator. So you might have heard or seen or felt horseflies. They're all in the family Humanipode, and it's the most species-rich family of blood-sucking insects. But it's kind of surprising because they let their presence be known. They're pretty noticeable in a lot of their different modes of sensory action. So we've been working in our lab for a number of years on horsefly phylogenetics, and this is a divergence time based on the whole family in beasts that's currently about to be submitted, where we try to make some links between horseflies and plants, since they're pollinators, and with their possible hosts, since the females do suck blood. And the really interesting thing in this tree, in my opinion, is that almost all the diversity in the whole family is centered right here in this area of the tree. So in my master's, I did a subfamily level phylogeny, where I used a couple more genes and a lot more taxa, but I still found almost no resolution, no support in one particular area of the tree where the vast majority of the species are found. So what I was really interested in doing is trying to figure out where that came from. Why did this group diversify in such a way? And it also diversified in a cosmopolitan way, unlike the subtending lineages, which are all neotropical. This most diverse lineage of horseflies is very common and very diverse worldwide. So that group is called the Tabanini. It's a tribe, but mostly it's composed of one huge genus, and the whole group has about 2,300 species. So that's more than half of all horsefly species. And they radiated relatively recently, in the last 50 million years. So for insects at least, that's a pretty recent radiation. So the timing of the radiation is pretty interesting because it's after the Cretaceous boundary and after birds and mammals, which are easier hosts for adult horseflies, also diversified. So horseflies have these semi-aquatic predatory larvae. So a lot of their biology is less well known since they are hidden in soil or in water, trying to eat other insects or even frogs and stuff like that. Although in this particular group, it's unusual because some of the larvae are arid tolerant, so they'll be in sandy areas. And the adults are pollinating and hematophagous. So there's kind of four big groups in the Tabanini. And I could have gone with clade A, clade B, but I decided to be a little bit nerdy and keep the genus names in there for you guys to learn. So the largest genus is Tabanus. There's over 1,300 species described, and many more undescribed, mainly in the tropics and Southeast Asia. And these are kind of the larger brown, more nondescript horseflies, but there's just a ton of species and a lot of diversity that's yet to be inscribed. The next largest genus is Humanipode, which used to be in a separate tribe. And Humanipode are more skinny and have patterned wings and have really really ornate eye patterns. So when I'm going to show you a lot of the stereotypical biodiversity images, but just keep an eye on the eyes of the horseflies because I think that's where there's a lot of diversity, and it's something I'm going to talk about later in the talk. The next large genus is pavitra, which has just these four eye bands, and they're mostly found in the far north Arctic areas. And then two kind of large-bodied fuzzy genera, Phylogenetic in the neotropics and Thyreoplectes in the old world tropics. These are bumblebee mimics. I wouldn't want to have them bite me, but they're really beautiful flies. So what I want to do with this study is kind of keep zooming in on this group and try to understand why did that diversify so quickly? Can we kind of get a better grip of the topology, get more support, more resolution in the phylogeny of this group, and also to try to figure out if those large genera should be placed within this huge genus Tabanus with greater certainty. So I'm going to first add more taxa to the question, and then try to add more markers with massively parallel sequencing. And then I'm going to talk about a little bit whether this phylogeny can be informative as to I-band evolution, and whether those I-bands, which give the flies more flexibility in vision, might have something to do or be contemporaneous with their radiation. So the first data set I'm going to talk about has 97 species, so well sampled at the genus level, the higher level. Of course, there's a lot more to do at the species level, since there's over 2,000 just in this group. And 43 tabanus with 5 genes, 3 nuclear protein coding genes, and mitochondrial CO1, and a big chunk of ribosomal 28S. So I'm kind of focusing on these two major groups, the tabanus group and the Humanipode group. So they're really different body plan, but it seems like this Humanipode group derived from within the more generalized tabanus group. So here's the tree, and I'll leave off the names. And the major thing you just got to know is the black nodes, the black taxa are in the genus Tabanus, and the gray taxa are in other genera. So as you can see, Tabanus is really complexly paraphyletic and includes about 15 other genera that total about 800 species. So Tabanus in the broad sense is a really massive genus of organisms. So all of these pictures here are in the genus Tabanus in the strict sense, and these are some examples of other genera in the group. So this phylogeny, this data set has really poor support in the backbone still, and there's some groups that are highly variable in their placement depending on the alignment and partitioning schemes. So I really hope to understand this phylogeny better with more taxa. So there's something really remarkable that's going on right in this major Tabanus radiation. And that can be seen in this splits tree, where at the base there's just a huge amount of conflicting nodes, conflicting information. So what happened there? I think one of the things that might have given rise to this huge diversification of horseflies is the diversity of I-band patterns. So there's other horsefly groups that have complex I patterns, but they pretty much either have it or don't. In this group, what's really interesting is that there's a lot of different morphologies, different shapes and patterns. So they can have bands versus stripes. They can have 1, 2, 3, or 4 bands. And then in this most derived group, they have really complex kind of like alien spacesuit eyes or something. So these I-band characters are map well within single groups within the phylogeny, but their pattern throughout the backbone of the tree is less well resolved and more unclear. But what is really neat is that the most complex I-band patterns, which is also in the most diverse sublineage, is the most derived within the tree. This is where the majority of the species can be found in a single group. So I'm really interested in trying to narrow down and see if we can really find a pattern that's left over in extant taxa of how these I-bands evolved. Was it really complex? Were they coming and going, or was it in stages where they became more complex as the lineages diversified? So I added hybrid enrichment data, and Al Lemon actually asked not to be on this talk, so he would have been, but he asked not to since they are redoing some of our taxa. So thanks a lot to the Florida State group, and I'm not forgetting about them. So we sent 10 of these horseflies to Florida for anchored hybrid enrichment, and got back data for 7 of them. And it's a probe kit that's designed just for Diptera based on transcriptome information. And these taxa were done in capture and sequenced along with 86 other fly taxa. And the final Tabanus data set has over 100,000 nucleotides. The filtering alignment done by Alan. So thanks a lot. And here is the resulting topology. So this here is the topology of just the 7 that we got anchored hybrid data for. And those blue taxa, you can find them in this larger tree. So this is with Raxamel, again, the black lineages are true Tabanus. So what was kind of surprising about this is that one of these taxa has a really long branch, whereas it didn't in the Sanger only 5-gene data set. So this blue one here is the same one here. So for some reason it has a really long branch. And also this group, which has one of the taxa with an anchored hybrid, really moved positions a lot. So I looked at the data a little bit, and it seems like there's a lot of multiple peaks, so we're glad that we're going to get a redo. But I just wanted to explore the anchored hybrid data a little bit more. So that I tried a number of sensitivity analyses with different taxon samplings, and I found that the backbone bootstrap support values do increase with anchored hybrid, which is really great because, you know, of those 100,000 nucleotides, only 6,000 of them have all the taxa, so it's a pretty sparse matrix in total. But there's two of the taxa that are really giving me problems in terms of switching around placements for unclear reasons. So I removed those just to see what happened, and I think we get those values are still really low, but for Tabanus, that's really great. You know, 41 bootstrap is way better than 3 or 9. So I'm pretty happy about the progress that we're getting in the phylogeny of this Tabanus group. And the anchored hybrid data has really high support. So I'm looking forward to exploring this more and getting a little bit more data. And I think it's really going to help us understand why this group diversified so quickly, when it did, and how it's different from most other blood-sucking insect lineages. So Tabanus, in the broad sense, includes 15 other genera, and adding more taxa to this phylogeny will help those taxa get placed in the phylogeny, but doesn't seem to do much for the overall backbone. I mean, even if I add 200 species, it's still less than 10% of the total diversity. But adding genes seems to include the backbone support, even for carefully selected representatives of major lineages. And the I-bands were present prior to the main Tabanian radiation, but the most complex I-bands are in the most derived taxa. So it seems like I-band diversification might be concurrent with the species diversification. And so in the future, I'd like to add more data, explore the data a little more with Partition Finder, try to get a Bayesian run with Exabase, since that might account a little bit better for the support values might be more clear with the spotty matrix. And also, I know this is going to come up, so in terms of species trees, I'm really interested in doing it. And I think what I'll have to do, since the lineage is so diverse, is to kind of pick one lineage that's less insane. So one that has like 10 species or 15 species, and see whether species tree data and concatenated data yield really different topologies, or if it's kind of close enough, good enough, and then go from there. But I am interested in doing species tree analyses, and also maybe using Cecilena's Bucky program to look at discordance just with the overall matrix. So with that, I'd like to thank my collaborators and lab mates and people who helped me with this tree, and those who lent specimens and images, and then I will take any questions. Thank you. Yeah. It's you. Yeah.
What was the function of the I-bands?
So it's not, you know, it's pretty well established that what they do is they filter out whatever light, whatever color that you see, is that they're filtering out that color. So let's go back to here. So the kind of idea is that if they are in like a dense tropical forest where it's very green, then the eye pattern will reflect back the green, so then darker colored hosts might stand out more against the background. So for instance, this one would be in more sandy environments, but why they start to become in very complex patterns is not so clear. I mean, you can imagine like if it was one part was green and one part was brown, that would give you some modularity, some flexibility. But the really complex ones are a little bit mysterious, but I think it still has something to do with host or mate finding. Any more questions? All right, thanks a lot, and thanks everybody for coming to the Phylogenetics and Systematics Symposium. Thank you.